So, Thomas, can you please tell me that role that the mitochondria play in the development of cancer?
They are a second living organism inside a cell. They actually have their own genetic material, and they are the organelle that breathes the air to make the energy. So, what Warburg found was that all normal cells have these organelles that take in the oxygen, and the cancer cells have a defect in that organelle. So they take in less oxygen and they produce lactic acid, which is a waste product of glucose fermentation. They call it lactic acid fermentation, and that proves clearly that these tumor cells are not getting energy through any kind of an oxidative mechanism. So as long as they have the fuel to continue their fermentation, they will survive. So all cancer cells, and every one that we've looked at, they have damage in their structure of that organelle. That means that all cancer cells, regardless of what you want to call them, are all fermenters. They're using an ancient fermentation pathway to survive. So that means that there's a very simple way to kill them. You just have to take away their fermentation energy and feed them things that they can't use. Fatty acids and ketone bodies are not fermentable. You need oxygen to get energy from ketone bodies, and the cancer cells don't use oxygen. So it's a very clear way to kill the majority of cancer cells. And then the amino acid is also, glutamine, is also the second fermentable fuel. Glutamine is the most abundant amino acid in our body. It's everywhere. So in order to, and besides, our immune system needs glutamine, our urea cycle needs glutamine, our gut cells all depend on glutamine. So if you go too aggressively on attacking glutamine, you also can cause metabolic disturbances in the body. So that's why we developed the press pulse theory of strategy. We can control glucose constantly because glucose is a non-essential metabolite. Our body does not need glucose. We can make glucose from within at very low concentrations. But the glutamine issue, you have to get the body into a ketotic state, and then we hit the glutamine just briefly, boom, wait, bing, so it's pulsed. It's like a pulsing. And that just, I have a few cancer cells here, they go down. Okay, then we let the glutamine come back, but not too many come back, just a little bit. Then they hit down, come back a little bit, hit down. And the next thing you know, gone. So again, you have to use knowledge of how do we kill cancer cells without harming the normal cells? And that's the strategy that we're developing as dosage, timing, and scheduling. That's where we are now. We're perfecting dosage, timing, and scheduling. And once we have those conditions perfected or modified, we'll be able to manage the majority of cancers with minimal effort. But we're right there at that point. So we do everything preclinically. In my lab, I have the best preclinical models of cancer: glioblastoma, systemic metastatic cancer, the very types of cancers that we see in humans. So we can practice on all this in the preclinical system. And once we see what works really well, we go right to the clinic with it. And my physician colleagues tell me, whoa, I did what you said. We're working out slightly different dosages and timing, but the results are really quite remarkable. So that's how it works. I develop the concepts, I test the preclinical system, and then we translate. There are very few people on the planet doing what I do. It's because I have the best models, and I have the concepts, and I have willing participants, physicians, that are willing to try this. Many of them realize that what they were doing in the past is not working. They're harming their patients. So all major cancers are similar in having reliance on a fermentation metabolism to survive. Without energy, nothing can grow. So where do these cells get their energy from? How do they get their energy? Once you understand how the cancer cell gets energy, then you can target that and they'll die. It's not that complicated. Oh, can we kill cancer cells if we take away their fermentation? Yes. What do they ferment? They ferment glucose and the amino acid glutamine. So if we take away glucose and glutamine, they should die. And the answer is yes, they will die. So, and they can't burn fatty acids or ketones, so the solution to the cancer problem becomes recognized. So why nobody is doing this? So, and in fact, when I say that, one of the problems, people think it's too simple. People want things to be complicated. So nothing can make a simple condition more complicated than saying that gene mutations are responsible for this disease, because it's infinitely complicated. And people think, oh yeah, so we're going to use all these strategies because cancer is such a complicated disease. And when I tell them, no, it's very simple. You can just kill it by taking away. I can't believe it. It's too simple. Warburg had the same problem. They said it was too simple. People don't want simplicity. They want complicated stuff when it comes to, I have to go through this and I have to go through that. So how do you break through this? I don't know. Conceptually and theoretically, it's not that complicated. But practically, to manage cancer, it does require knowledge about how you will, in fact, lower those two fuels, which then becomes responsible of the patient. It's funny to me. People sometimes have this attitude that I have this terrible disease. Maybe it was caused by God or something. Maybe I did some bad things. So I'm going to take chemo and radiation, and I'm going to suffer. And because I suffer, it should get rid of my cancer, because suffering is kind of like the penalty you have to pay or something along these lines. But then you come along and say, well, the concept is not that complicated. But when you stop eating for a week, you feel like you're really suffering, because our brains are addicted to glucose. So once you get through the glucose addiction, actually the management of the cancer becomes so much better. So we have to transfer this idea of painful suffering from toxic poisons into a self-directed reduction of food intake, or taking diets that lower glucose and elevate ketone bodies, so the cancer can be better managed.
So what should we eat more of, and what should we eat less of?
Well, I think the basic thing is eat less of everything you might be eating, because the best way, but when you tell people to get rid of their cancer that you have to stop eating for a month, they think you're crazy. This guy must be nuts. Who can stop eating for a month? Many people feel that if they stop eating for 3 days, they're going to be dead. This is how little knowledge we have of our incredible capabilities of living on the planet without eating. But we're trying to make water-only fasting, which will kill cancer cells quite effectively, more reasonable with a diet that is palatable, nutritious, and will lower the blood sugar and elevate the ketones. So we developed the glucose ketone index calculator to allow people to know if they're in a zone for killing cancer cells. So they always ask me, what can I eat? And I say, well, eat anything you want, and then check your GKI, the glucose ketone index. If it makes the glucose ketone index high, don't eat that. So only every person will find their balanced amount they should eat and what they should eat to keep the ratio down, and that number will tell you that you're killing cancer cells. So how do I know if it's working? It always works when you're in that zone. Oh, I can't get into that zone. I've done everything I can. I can't get down to 2.0 or below. Well, what do you want? I'm eating this and I'm eating that. I said, well, don't eat any of that. Just drink water and see, it went right down. Okay, now add back the foods that will keep you in that zone without throwing you out of it. So it's an experiment on each individual. And they experiment what they can and cannot eat, how much they should eat, and how much they can't eat. So that's all worked out for individual people, and they keep their records. They keep a log of all this. So just like a diabetic, you can't eat that, can't eat this. I'm keeping my blood sugar low. So it all works from the individual.
So tell me, why is it that the work that Otto Warburg did, that we didn't continue in that path?
Otto Warburg knew what the origin of cancer was. The problem is his quantification of lactic acid and oxygen consumption was misunderstood. This is an extremely important point. I'm preparing a publication to show how Otto Warburg and the entire field of cancer at that time were misinterpreting their observations. So we just have taken Warburg's concept, polished it all up, and now can explain clearly what the origin of cancer is. Once the cancer field comes to understand this, then they have no choice but to target glucose and glutamine simultaneously. It becomes the obvious and clear pathway for managing all cancers. And this is another thing that gets everybody all upset. They think breast cancer is different from colon cancer, different from brain. They're all very, very similar. They need those fuels to grow. They can't use oxygen. That's the similarity. They're all genetically different. They look different under the microscope, but they're all similar. They need fermentation, target the fermentation, and you can manage this disease. So that's the bottom. When you get enough people that respond well, healthy, didn't lose their hair, didn't get all this toxicity, and their cancer, very deadly type of a cancer, goes away, more and more people will want to do that. And it just takes time for the field to come to recognize this. If you're doing radiation for brain cancer and you know that it's actually contributing to the death of the patient, and someone clearly told you that and how it's happening, how do you feel continuing to do that? It becomes a moral question. Is it moral to treat somebody with a therapy that you know will kill them? Before you didn't know that. You thought you were doing good. Now the evidence says by doing that you're going to kill your patient. Well, I'm going to do it anyway. That's immoral. So then they have to deal with the moral question.
What are the reactions when you talk about this? I mean, among the oncologists?
Well, they don't want to hear about it. It's like a religion, okay? How hard would it be to tell a person who is a devout, practices Islam devoutly, and then you tell him Judaism is the correct religion? Do you think that guy's going to change his religion? No. It's a dogma. It's a system belief. So the idea that cancer is a genetic disease has become a dogmatic view in the brain. It can't be anything other. I was trained from high school, college, medical school. All my professors told me cancer is a genetic disease. The National Institutes of Health says it's a genetic disease. Therefore, I feel justified doing what I'm doing. Even though I see this other, I can't accept it, even if it's right. I can't accept it because I'm dogmatically brainwashed into thinking, I'm indoctrinated to think this is what it is. So you have to break. I find indoctrination is more powerful than most genes. I mean, when your brain is solidified into a way of thinking, some people would rather die than change their political belief or religious belief or their belief in a certain. They're going to fight tooth and nail to keep their dogmatic view. But in cancer, you're dealing with all these people that are dying and suffering. So eventually, your dogmatic view is going to have to change if you want to make a difference in this world in treating this particular disease. So it's a treatable disease without toxicity, quite cost effective. Get over it. It's going to happen. It's only if you're doing all that, it's only you're just holding back the lives of these people. You're preventing people from living a better life. You're standing in the way of progress. And the progress we're making is just simply going back and modifying what was already known and bringing it into a more clear perspective. So the clarity of what Warburg did, we've cleared up a lot of the misinformation and the misunderstanding. His theory was correct. The output of explaining his theory was not correct. So we've corrected that. Now we know, and there should be no reason to do anything other than focusing on this because it works and people can live a lot longer if not having complete resolution.
What about chemotherapy and radiation? Doesn't it do any good?
Well, it obviously had some. I mean, we have what we call a lot of cancer survivors. So we have millions of cancer survivors who have taken radiation and chemo, and by that they're alive longer. The problem is that when your body is subjected to chemo and radiation, the cancer may not have survived, but you've damaged significantly many organ systems. So you suffer from cardiovascular disease. And one of the neuropsychiatric impact of being subjected to those chemicals is quite impactful on a lot of people. Hormonal, digestive problems. In other words, you're confronted with a new series of health issues. The cancer may not be there, but your body has been damaged, and now you become a patient for these different kinds of problems that came as the result of being exposed to highly toxic chemicals and approaches. So metabolic therapy is nice because not only do you get rid of the cancer, you also allow the body to heal itself and become even more healthy in a lot of different ways. So you avoid all the toxic effects that you would get from those standards. So yeah, I mean, they work for a lot of people, but you pay a significant price Your body pays a significant price in damage from radiation and chemo. But everybody says, oh, I'm so happy I can deal with digestive issues, with neuropsychoses. I'm so happy I'm still alive. But why don't we say, well, we can keep you alive better and you won't have to have any of that. Everybody would want to do that. Why would they want to subject their body to any kind of a toxin if they knew there was another way to do it? And then all of a sudden we'll look back in 100 years and say, I can't believe we did all that crazy stuff to those cancer patients. You look back and you say, what profound lack of knowledge when there was a guy, Warburg, who actually had it pegged. The problem is nobody hears about it because they're not being published. So what I do is I'm writing case reports on individuals and showing all the data, and look what happens to this patient, look what happens, but they're not, oh, well, you need a big clinical trial before I'll believe it. So you don't believe that all these people could be real because they weren't part of a trial. Now, the problem with clinical trials is you have a drug and no drug, okay? And then you do switch over. We double switch over. Well, every person in metabolic therapy is getting a kind of a diet. It's hard to say what are you getting, and then switching back and forth. Is it moral to take a person who's really managed cancer and switch them back to a different kind of a drug when they're already managed? You can't do that. So we have to redesign the way clinical trials are done for metabolic therapy. You cannot use the standard pharmaceutical approach for doing clinical. I can't believe it unless we do this clinical trial dictated by pharmaceutical companies. No. You got to get rid of that. You have a different, we look at survivability. How long does the patient survive relative to what the field has shown? We have so many survival curves for all these different cancers. And if we can push that down double, 3, 4 times survival, you're telling me that we can't use that data because it wasn't done by a traditional type of clinical trial? I mean, this is nonsense, complete nonsense. So you have to then reestablish how we're going to do new clinical trials and then will they use it, even after you do all this? And the answer is we don't know. So the origin of cancer is mitochondrial damage. How does that happen? From any number of different things in your own body or in the environment, all leading down the same path. So we put it all together. I did all this research. I tracked down every one of these risk factors and find out what did they do. They all damage the mitochondria. Not a mystery. If you read the literature and you understand the scientific literature, what I'm saying is not mysterious. It's there in the literature. Just people don't know about it. I try to publish the books. I try to do everything. I mean, what are you going to do? Grab somebody's head? You mean? Okay. So they don't recognize cancer as a metabolic disease. When you realize that it's a metabolic disease and the mutations in the cancer cell prevent it from being, they're locked into a very tight fermentation. If you can't ferment, they're going to be dead, and they don't have the ability to adapt like the normal cells have. It all goes back to evolutionary biology. If you don't understand evolutionary biology, you do very stupid things, okay? Like immunotherapy, very stupid. So, and you do all these crazy things because you don't understand evolutionary biology. What are you supposed to do? You know, and unfortunately the field doesn't, they look at you like, you might as well be talking to a plug socket because they don't even know what the hell you're talking about. So, I'm telling you, it's terrible, right? All these guys, they talk like they know what they're doing. You know, no, Jack, if they knew what they're doing, why are they doing this crazy stuff to these poor cancer patients? So you see how it all comes together? It all comes together because I understand evolutionary biology. And when you understand that, you know how to, that's so many nice ways we can kill cancer cells without toxicity. You just exploit their weakness, their metabolic weakness. And when you realize how many ways we can kill cancer cells without toxicity, it's just so almost infinite once you understand the system. So it just takes time. That's another thing, metastasis, which is the singular most complicating factor of cancer. Because if all tumors grew in place, we would just cut your arm off or cut whatever, and it's gone. But it spreads. So how does it spread? What's going on with the spread? So what we discovered was an interesting kind of cancer in the mouse. It came out of the brain spontaneously, but when we put it in the flank, it spread like wildfire throughout the whole body of the mouse. And then it took us 10 years of vigorous, constant research. What kind of a cell? What is that cell? What kind of a cell is this thing that spreads all over the body? Because this is what is responsible for the majority of cancer deaths. So we found out that it was a biological, it was a macrophage, a microglial cell, which is derived from macrophages in the brain. It's a localized thing. So a macrophage, huh? Wow, macrophages are the only cell that can move in and out of our tissues. So we have resident macrophages in all of our cells. They're residents, they're like a police department. And then we have white blood cells circulating through our bloodstream, and that's like the army. So when you have a stab wound, white blood cells come out of the blood and they transform into these macrophages, and they're there to kill bacteria, clean up the wound. They're very fusogenic. They fuse together to work and heal the wound. In the cancer, what we have is that there's an inflammatory group of cells, like chronic inflammation, hypoxia, whatever, causing what looks like a wound to the body. So the body sends in the immune system. If the local guys can't handle it, the immune system, and they throw out growth factors and cytokines to heal the wound. And what that does to a cancer cell, like a stem cell, that doesn't have the capacity, it just grows in place. It makes them grow more. So these immune cells fuse together with these other kinds of destabilized cancer cells that have mitochondrial abnormality, and they're growing but they can't spread. They're not. But this cell has the genetic apparatus to already move in and out of tissue. So when that cell fuses with this other cell, now you get destabilized mitochondria in a cell that's programmed so it becomes a rogue macrophage, moving in and out of the cells. Wow, this is like unbelievable. So how do I know this? Why would I tell you this if I didn't know what I was talking about? So we went through every major cancer that we know of and examined genetic readouts of macrophage products. Every major human cancer has all of these characteristics of macrophages. So they all started from this procedure. The late John Powlick at Yale University was screaming at the top of his lungs, this is a fusion hybridization between macrophages and cancer stem cells, producing this metastatic cell that can now readily spread throughout the body. So after we made this discovery that nobody believes because they're locked into this mindless genetic thinking. So first thing you say, what do macrophages eat? What's their fuel? What do they do? They'll eat anything, but they gobble up stuff and they bring it to their lysosome and then they can get energy from what they do. But one of their prime fuels is glutamine. Macrophages in the immune system are driven by glutamine. Aha! So then we found they're fermenting the damn glutamine. So now we know how to kill metastatic cancer cells. You take away the glucose and the glutamine, and we've shown this. So everything comes back to knowing the biology of the problem and what cells are eating, what they eat and what their biology is, and how do you put all that together. And you can come up with a clear idea of what cancer is and how to manage it very successfully. What's wrong with this? Why can't anybody understand this? Well, the money issue is something, but eventually the people want to live. And so I've just defined to you the strategy, and believe me, it's going to happen, because we can't keep our head in the sand for too long. It's one of the great tragedies in the history of medicine. And sometimes you have to take a little levity, and you have to realize how incredibly disconnected we are from knowing how to do things when the solution is staring you in the face and you choose not to use it. That's tragic. That's tragic. So. Yeah, I mean, what am I going to do? But I see it, and I have a voice, and that voice is in peer-reviewed scientific publications, participation on journals, editorial boards. I have the knowledge. I do the research. I can communicate the research. We have patients that are surviving. This is evidence. So I'm not some lunatic. I have hard evidence that supports everything that I'm saying. Let the public choose to accept it or not. So there has to be some public outrage. It's never going to come from the hospitals. It's not going to come from the pharmaceutical companies. It's got to come from the people. Just like Dr. Rao was saying. The people must be educated to know that there is a viable, scientifically established alternative to this crazy stuff that they're doing at the top medical schools. And once the people realize that, you're going to start seeing things happen. Because nobody wants to go through this kind of crazy stuff. So when they say we've made big progress in stopping cancer, they always go back to the 1991 when cancer was on a really rocket rise because of the smoking. So Now they stop smoking, cancer goes down. So today we're seeing a rise of cancer, but they say, look how much better we did. Well, we stopped smoking. All the major advances in cancer come from prevention, not from treatment. Treatment has been an abysmal failure for the most part. So it's changing in behavior. So if you change your diet and lifestyle, and you can certainly manage cancer, prevent it. But I don't think people want to change their diet and lifestyle until they get cancer. Once they get cancer, then they know that they're going to have to do something. So then that's when it happens. But yeah, the anti-smoking campaign is largely responsible for the perceived advances in cancer. It's all prevention. Very little, very, very little to do with treatments or diagnosis and all this kind of stuff. So, and they already know some of these diagnostic breast mammograms, they can put people at risk for further spread. Biopsies and all this kind of crazy stuff that they're doing is, why are you doing a biopsy? Oh, I have to know if you have stage 1 or stage 2 or stage 3. Well, if you had stage 3, well, you would never want to stick it. And if you have stage 1, you stick it, it could become a stage 2 or 3. Why are you doing that? Well, we do it because we were trained to do it. Would you ever think about, oh, I didn't know about that. You read the damn literature, for Christ's sake. These people don't read the scientific literature to show what they're doing to put these people at risk. I can't tell you how many oncologists. I did a big interview with Peter Attia. He said, Tom, you shouldn't be saying things like this. It's not science. I said, what do you mean, Peter? I said, for Christ's sake, do you read the literature? Well, I didn't see those articles. Well, I'll share them with you. I have a whole slug of them, published in the top medical journals. Biopsies spread cancer. So what are you doing that for? You know, this is nuts. In so many levels, it's complete nuts. And it's not getting better, it's getting worse. So it'd be one thing to say, Seyfried, you know, cancer's already, we're doing really well. You know, I think, you know, let's stay the course. Are you kidding me? It's taken over. It's replacing heart disease as the #1 killer of people on the planet. Millions and millions of people are dying from cancer. And it doesn't have to happen.